Hello friends, how are you doing? And I hope you really all are having a great time. Today we are going to discuss questions that on the chapter Biomolecules. We'll move on with the first question. What is an apoenzyme? And all these are 1 mark questions first of all, right? So an apoenzyme is a protein part, okay, an apoenzyme is a protein part that is attached to the coenzyme, right? So an apoenzyme is a protein part that is attached to the coenzyme in order to make it active, right? So an apoenzyme is a protein part that is attached to the coenzyme in order to make it active, and this is what is an apoenzyme, right? We move on to the next question, that what is ligase? From the word ase itself, you know that this is an enzyme, right? What is this? This is an enzyme. So the ligases, okay, are group of enzymes. The ligases are group of enzymes that join or form the phosphodiester bond in nucleic acid, that forms phosphodiester bond in nucleic acids. Right? The ligases are a group of enzymes that form the phosphodiester bond, in the nucleic acids. Right. Monosaccharides are called reducing sugars, but disaccharides are not. Why? Write an example of each type of sugar. Right. Now reducing sugars are sugars, which can react, which can reduce other molecules. So they need to have the reacting group. They need to have that group, which will attach and reduce or react to other substances. And yes, these reducing sugars have that aldehyde or the ketone group that will react. Okay. So monosaccharides, for example, the glucose and all are, the reducing sugars, right? Monosaccharides, like for example glucose, okay, like for example, okay. So monosaccharides like for example glucose will be monosaccharides like for example glucose will be acting, will be doing what? They will be have the reducing group like the aldehyde, or the ketone group. So, monosaccharides like for example glucose have the reducing group like the aldehyde or the ketone group, and hence they react, and hence they react with other substances, okay, thus called as the reducing sugar, thus called as reducing sugar, okay, thus called as a reducing sugar. So, I have already stated example glucose, and okay, are called reducing sugars. Disaccharides are not. Disaccharides, for example sucrose, they do not have the free aldehyde or ketone group, and hence they are non-reducing sugars, okay. Disaccharides, for example sucrose, does not have, okay, so disaccharides for example sucrose does not have the reducing group, does not have the reducing group like free aldehyde and ketones, like free aldehyde and ketones, and hence are non-reducing, are non-reducing. Disaccharides for example sucrose does not have the reducing group like free aldehyde and ketones, and hence are called as non-reducing sugars, okay. We will now go on to the next question. What are the two advantages of storing carbohydrate in the form of polysaccharide? Advantages. Now, first of all, when we are storing polysaccharides for example starch and glycogen, polysaccharides like for example, right, like for example starch and glycogen, right. So, polysaccharides like for example starch and glycogen store food and hence can be used as reservoir of food, okay, and hence can be used as food reservoir, okay, and hence can be used as food reservoir. Also what we see, the polysaccharides, the polysaccharides, okay, are formed by the removal of water molecules, by the removal of water molecules. So what happens, you know, it makes them condense, it makes them, you know, occupy less space, hence condenses the molecule, and also water is not utilized, okay, and also water is not utilized. Also the polysaccharides are formed by the removal of water molecules, hence condenses the molecule, and also the water is not utilized, right. So now we move on to the next question. What is the name given to that part of the enzyme where the catalytic work is carried out? Yes, that part is called as the active site. Name given to that part of the enzyme where catalytic, okay, where catalytic work is carried out is called as the active site, is called as the active site of the enzyme. Now you see, you know, that at this active site the substrate molecule will come and bind to the enzyme, okay. At this active site, we are doing the fifth question, at this active site the enzyme, at this active site the substrate molecule, okay, the substrate molecule will come and bind to the enzyme, okay. We now move on to the next question. How do animals digest cellulose? Important question. Do we have the enzyme to digest cellulose? No, absolutely no, because the cellulose, yes, it is a polysaccharide, it has a beta glycosidic bond, and we cannot break beta glycosidic bond, so we cannot digest cellulose. But animals, yes, the ruminant animals have a compound stomach. The ruminant animals have a compound stomach for the digestion of cellulose, compound stomach for digestion of cellulose. Now what happens here, we have the symbiotic microorganisms, for example the Rhinococcus in the ruminant animals, which secretes the enzyme cellulase which digests cellulose. So have Rhinococcus enzyme, Rhinococcus enzyme, okay, have Rhinococcus enzyme which secretes the enzyme, have Rhinococcus, you know, that's the name of the microorganism which secretes cellulase for cellulose digestion, right. Similarly, we will see that even termites have trichonympha. Okay, termites have trichonympha, which again secretes cellulase for the digestion, helping in digestion of cellulose, helping in digestion of cellulose. Termites have trichonympha which help in the digestion of cellulose. We move on to the next question. What is contact catalysis? What is contact catalysis? Now, in contact catalysis, right, the substrate comes closer, okay. In contact catalysis, what happens? The substrate comes closer to the active site of the enzyme, to the active site of the enzyme, right. So, in contact catalysis, the substrate comes closer to the active site of the enzyme where the reaction would take place, okay, where the reaction would take place. So, what is happening here? Basically, the substrate is coming in contact with the catalysis over here, and what is the catalyst over here? Catalysis over here. The catalysis is the reaction and the catalyst is the enzyme. So, it is coming in contact with the active site of the enzyme, and this is what we mean by the contact catalysis. What is the difference between enzyme and catalyst? Absolutely wonderful. Enzymes and catalyst. The enzymes are mostly proteinaceous particles, mostly proteinaceous, okay, and these are mostly inorganic particles or whatever substances, mostly are inorganic. In nature. So, the enzymes are mostly proteinaceous, however, the catalysts are mostly inorganic in nature. The enzymes work within the living body, within the living body. They can work both in the living as well as the non-living environment, right? Can work both in living and non-living environment, right? So, they can work within the living body, and these can work both in the living as well as the non-living environment. The enzymes, the enzymes work on a specific action or reaction, work on a specific part. Now, these work, the catalyst, a single catalyst can carry out many reactions, okay. These can work on many actions. These can work on many reactions, okay. We will now move on to the next question, that yes, In the stomach and small intestine, the enzymes for digestion of protein are secreted in an inactive form. Why? See, when we talk about the stomach, in the stomach, what is all is secreted. First of all, there is pepsinogen secreted in an inactive form. Okay, the pepsinogen is secreted in an inactive form, and even the prorenin, right? So pepsinogen in the presence of HCl will form pepsin, and this also in the presence of HCl will form the renin, right? This pepsin will act on the proteins. The pepsin will act on proteins and convert it into peptones and proteoses. Peptones and proteoses. And similarly, in the intestine, what do we see? Intestine, what do we see? That the trypsin and the chymotrypsin. So first of all, there is an inactive enzyme called the trypsinogen, okay, of the pancreatic juice, which is activated by the presence of the enzyme called the enterokinase, which belongs to the intestinal juice, and in this presence it will form the trypsin. This trypsin will again act on the proteins and convert it into peptones and proteoses, and also we have the chymotrypsinogen. This chymotrypsinogen will be acted upon by trypsin. Now this trypsin has an autocatalytic action. Once small amount of trypsin is formed with the help of this enterokinase, it will, this trypsinogen will get converted in the presence of that trypsin only. Okay, the chymotrypsinogen will form the chymotrypsin. Now what we see that these enzymes are activated, are secreted in the inactive form. Why? Because see, this is, this will be hydrolyzed or metabolized, right? Till the time, there is acidity, it cannot work. It cannot work, and only, when there is food in the body, it cannot work all the time also. So only when there is food and there is acidic environment will work. So when the HCl is secreted by the oxyntic or parietal cells of the gastric gland, that will work on this pepsinogen. At that time the food is also there, and that helps in the formation of pepsin. Also the same thing applies here. It cannot work all the time. Only when the alkaline juice is there, then only it will work. So this in the presence of enterokinase forms the trypsin, right? We now move on to the next question. Why doesn't oil dissolve in water? Give a scientific explanation, right? Water. Now water, what does it consist of? Water consists of hydrogen bonds. Okay, water consists of hydrogen bonds. So anything which has polarity, okay, anything which has polarity and can form hydrogen bond and can form hydrogen bond, okay, anything which has polarity and can form hydrogen bond will so anything which has polarity and can form hydrogen bond with, okay, and can form hydrogen bond with water will dissolve in water, okay, will dissolve in water. However, oil is a non-polar compound, Oil is a non-polar compound, cannot form hydrogen bond, cannot form hydrogen bond with water, hence does not dissolve in water, does not dissolve, okay, hence does not dissolve in water, okay, right? Can you see this till here? Okay friends, so we have come to the end of these 10 questions and discussion. Hope you followed it. Get back, revise this, any doubt you can always get back to me. The time we meet again, a very goodbye from this side. Thank you.